Next area of microbiology is geomicrobiology. Geomicrobiology involves the study of microorganisms that are involved in geological processes such as withering of rocks, soil, and degradation of minerals and fossil fuels. So these microorganisms are very useful for the extraction of metals or extraction of minerals from the different parts of earth like soil and rocks. Extraction of metals by microorganisms is called as microbial leaching of metals. So, microbiologists have isolated many microorganisms, especially bacteria, from rocks and soil and mine. So, these bacteria are basically very useful in the various mining activities. These microorganisms are chemolithotrophs. So these organisms are not ordinary microorganisms like these microorganisms are not general microorganisms. For example, Escherichia coli, which is present in our intestine, lactic acid bacteria, which are present in curd. All these are general category microorganisms which require normal growth conditions. But chemolithotrophs are the organisms which require or which survive in extreme environmental conditions. So, in mines, the conditions are not normal. We call these conditions as extreme environmental conditions where there may be deficiency of oxygen, there is presence of different types of minerals, deficiency of different types of nutrients, then acidic conditions. So all these extreme conditions are existing. Still, these microorganisms are able to grow in that environment. These are called as chemolithotrophs. These microorganisms use or they generate energy from the oxidation of inorganic substances. For example, they generate energy from oxidation of iron, that is from conversion of Fe2 plus to Fe3 plus. So this is the unique characteristic. In case of most of the aerobic bacteria, they use they use oxygen as a terminal electron acceptor, whereas these bacteria use metal ions as electron acceptor in electron transport chain. So rocks are the rich sources of various minerals, but these metals or minerals are present in complex form. So their extraction from that complex form, so they can be released or they can be freed from those complex rock form or soil form into simple form, or they are made extractable by means of microorganisms. Smelting. It is a traditional process for the processing of ores. Ores are the impure form of metals which are present in the mine. So this ore, it contains the metal, but it is present in a complex form along with lots of impurities. So there is a traditional method of leaching of metals called as smelting. It involves heating and melting of ores to extract the metals which it contains. However, this method leads to the consumption of lots of energy, and this method leads to pollution also. The chemolithotrophs or autotrophic bacteria, they are capable of improving this situation. That means extraction of metals is possible by microorganisms, and this is how we can avoid the use of huge amount of energy. We can save huge amount of energy that we spend in smelting process, and use of microorganisms will also be an eco-friendly method, as there is no pollution. There will be no pollution because of this technique. These microorganisms improve the recovery of metals from an ore and does not cause pollution of atmosphere. The autotrophic bacteria, when grown in presence of metal ores, cause oxidation of ore and subsequent precipitation of metals. And then these precipitated metals can be easily separated from other impurities. Most widely used bacteria in the microbial leaching of metals are Thiobacillus ferroxidans, which is a gram-negative motile rod-shaped bacterium. It is a chemolithotrop which derives its energy from oxidation of iron, that is from conversion of Fe2 plus to Fe3 plus, and carbon from carbon dioxide under acidic conditions. The metals such as copper, uranium, and many more can be extracted or leached from low-grade ores using Thiobacillus ferroxidans. In case of leaching of copper, copper is present in the form of low-grade ores such as copper sulfide, which is called as covellite, or chalcopyrite. The waste ores are often dumped in the form of heaps from which the metal can be extracted. An acid leach solution. Now we'll see an example of microbial leaching of copper metal. So for this leaching process, it requires following things. First one is an acid leach solution that contains this bacteria, Thiobacillus ferroxidans. Second, Fe3 plus means ferric ions and sulfate ions, SO4 2 minus, which forms sulfuric acid in water. So this solution is sprinkled on the metal containing ore heap. Fe3 ions oxidize cuprous ions that results into formation of Cu2 plus means cupric ions. This is oxidation process. Then the sulfuric acid solubilizes the copper sulfate, that is CuSO4. This is metal impregnated solution that contains copper sulfate in the form of Cu2 plus and SO4 2 plus ions, and ferrous ions, Fe2 plus ions. Next, from this solution, copper, that is Cu2 plus, can be precipitated with the help of waste ion, that is Fe0, to give ferrous sulfate and elemental copper, that is Cu0. Cu0 means it is the precipitate of pure copper metal. Thus, after the removal of copper, the remaining solution goes to an open pond which is called as oxidation pond, and this oxidation pond, in presence of oxygen from air, Thiobacillus ferroxidans, it causes the oxidation of ferrous to ferric, which is the source of energy for the organism. So this solution with ferric ions and sulfate in the form of H2SO4 is finally pumped back onto the ore pile. That is the first step, and this is how the cycle continues. Let us understand this in the form of a diagram. So there is an oxidation pond in which bacteria, Thiobacillus peroxidans, are there along with air. So this Thiobacillus peroxidans along with other nutrients contains Fe2 plus, means ferric ferrous ions, ferrous. So these bacteria, they will grow in this oxidation pond, and while growing these bacteria will generate energy by converting Fe2 plus to Fe3 plus, that is ferric ions. Then these ferric ion containing solution, which is called as acid leach solution, that contains ferric ions and sulfate ions in the form of sulfuric acid. So ferric ions and sulfuric acid are formed due to the growth of Thiobacillus peroxidans. This solution is then sprinkled over the large heap म्हणजे ढिगारा, large heap of copper ores. These copper ores or the rocks containing copper are collected from mines. So this heap of low grade copper ores dumped at copper mines, they are sprinkled with this acid leach solution. As a result of that, this acid leach solution, which contains Fe3 plus, this Fe3 plus oxidizes this copper to Cu2 plus, and this Cu2 plus then combines with H2SO4 to form copper sulfate. So the function of H2SO4 is to solubilize copper ions in the form of copper sulfate. This is then pumped to a separate tank, which is called as pregnant solution, which contains metal bearing, which is a metal bearing solution that contains copper sulfate and ferrous ion. This copper sulfate, now from this copper sulfate we need to separate copper and sulfate. So this is mediated by the addition of West ion. So this West ion is present in the form of Fe0. This Fe0 will combine with CuSO4, and there will be formation of Cu0 plus FeSO4. Cu0 is nothing but the precipitated copper. So this precipitated copper is then separated from this solution. That means this precipitate of copper is insoluble. So it is separated from the rest of the liquid. This liquid contains Fe2 plus ions. So these Fe2 plus ions are good nutritive source for Thiobacillus peroxidans. So this solution will then go to the oxidation pond, where again Thiobacillus peroxidans regenerate Fe3 ions, which are required for the formation of or the release of copper. This is how the cycle of leaching it continues. Just for the sake of your information, you can see this is the largest mines for the ore of copper, that is Escondida in Chile, that is in South America, is the biggest copper producer in the world, accounting for 5% of global production. Bacteria such as Thiobacillus peroxidans, Thiobacillus thioxidans, Sulfolobus acido-caldaricus can oxidize sulfides to sulfate. This is a very important property that can be used for purification of charcoal. So this bacteria can be used for the treatment of charcoals, treatment of coal, because this coal contains sulfides, and these sulfides may lead to the pollution, means the burning of coal containing sulfide, it leads to the problems such as acid rains. So the isolation of these types of microorganisms may lead to cost efficient method to desulfurize coal, removal of that sulfide or conversion of sulfide to sulfate. This is called as desulfurification. This makes possible to use coal as a major or more attractive energy source, and also helps to solve the problem of acid rain. One of the major environmental problems of industrialized region of the world is acid rain. The principal sources of acid rain in atmosphere are oxides of sulfur and nitrogen. So these oxides of sulfur and nitrogen are generated due to burning of sulfur containing coal. So air movements, they will distribute these oxides, and therefore the harmful effects can be found at greater distances from the source of pollution. So this is really not good for the life of aquatic animals and forest lands. So the burning of coal in different power generation plants and factories generates sulfur dioxide. The emission comes from the sulfur found in coal. And automobile and fossil power plants are principal sources of nitrogen oxide. Coal burning steam generation plants used to produce electricity. Thus, the removal of sulfur dioxide from coal in an economical manner would help to solve the problem of sulfur dioxide in the atmosphere. So microorganisms are the solutions to this problem. Bacteria such as Thiobacillus ferroxidans, thioxidans, and Sulfolobus acidocaldarius can oxidize sulfides to sulfates as mentioned earlier. Another application of microorganisms in geological processes is in petroleum industry. So there are several reservoir sites of petroleum in the soil. So microorganisms can be used to detect these storages present in soil. These microorganisms are using hydrocarbons, means petroleum products, for their growth. So if these microorganisms are introduced into such habitats, the test system will show that the microorganisms are growing. So why microorganisms are growing there? Because there is presence of hydrocarbon. So presence of hydrocarbon means there is presence of petroleum product. This is the concept of a biosensor. That means microorganisms are used for the detection of petroleum reservoirs. Microorganisms, biological things which are sensing, which are indicating the presence of petroleum products. So this is an excellent example of biosensor, microorganisms being used as biosensors. These microorganisms are also useful in petroleum recovery. Now, this is a very interesting topic in several oil industries, or in several petroleum product extraction industries which are there in UAE, or the several other countries. They are making use of this microbial technique for the extraction of petroleum products from oil wells. So when an oil well is drilled, bacteria are injected into these oil deposits. So these bacteria, they produce acid, and this acid is responsible for the dissolving of rock. So oil or the petroleum products are trapped in complex form in rocks. These microorganisms, with the help of production of acids, they allow the release of oils or petroleum products. So release the oil and also increases the pressure due to production of gas, and that helps the to improve the flow of petroleum products in oil wells. Microorganisms can also decrease the viscosity of oil, and that is responsible for improvement in the rate of flow to the surface. This is a very important and widely used application of microorganisms in petroleum industry. So this is a beneficial part. There are some microorganisms that are harmful. For example, Desulfovibrio. This organism is responsible for the corrosion of iron pipes. This is a major problem in oil industry. Few microorganisms are also known for the degradation of petroleum products. This part we have already studied in my first lecture, that microorganisms are very useful for the treatment of oil spills. So the incident of Mauritius oil spill we have already discussed. That means these are the bacteria which are used to decrease the pollution, which are used to clean up the oil. The oil spill area is inoculated with these microorganisms that can degrade this oil, because wreckage, due to the wreckage of oil reservoirs, the oil is released, and which is not good for the life of aquatic animals. So in order to prevent that, in order to treat that oil spills, microorganisms such as Pseudomonas putida has ability to metabolize these oils, or they use this oil as a source of carbon and energy, and it brings the degradation of that oil, and this is how it helps in preventing the development of pollution.